right, this is the first lecture in Core 2. Just to reiterate, you should have the study guide in front of you so you can take notes within the study guide. So this is the first part. And as we go, what I'm going to do is always back up and show you what I'm going to talk about in the short lectures. And then I will go through each thing a little more in depth. Okay, so this lecture is on the conditions of the young Earth that would make the origins of life possible. And so it's basically like how did the Earth, or how was the Earth able to produce the first single cells? And if you remember from Core 1, remember that cells are made up of phospholipid bilayers that have basically water on the inside of them and water on the outside, and the water that's on the inside has molecules. But the other thing that makes them life is that they have self-replicating molecules, therefore the organism itself or the cell itself can reproduce. All right, so this is called the four-stage hypothesis. And in order to produce the first single cells, you need abiotic synthesis of small organic molecules. And then you're going to make polymers and package the polymers in that phospholipid bilayer. And then you have to have those self-replicating molecules. So ultimately, that will create a self-replicating cell. All right? So let's just go through this a little bit. So abiotic synthesis of small organic molecules, they think could have happened, at least several people have done experiments with the conditions of the young Earth. So just to kind of go over what those were, that is, there was, they thought there was a little amount of oxygen. That is oxygen gas. There was oxygen as a molecule, but not oxygen or O2 that existed freely within the atmosphere. Therefore, there could have been little catabolism, that is, like the breakdown of organic molecules requires oxidation, generally speaking. And photosynthesis, of course, would not be prevalent, and photosynthesis producing oxygen was not ultimately producing oxygen. So which came first? Not clear, but it was clear that photosynthesis was not prevalent during that time. Lightning, or electrical impulses in the atmosphere, volcanic activity, and much higher UV light because there was less of an atmosphere just in general. All right, so several people over the history, like O'Parent and Haldane were one group, Miller and Urey were another group a little bit later. They tried to, well, first of all, O'Parent and Haldane just tried to theorize about what the conditions were, and then Urey and Miller actually did experiments on that. And what they did was they put, they tried to, create a closed system. Of course, no system is completely closed, but what they did was they put molecules that would be, or I should say atoms that were part of schnapps in here. In other words, basically this is water and creating water vapor, which would then mix with CH2 or methane, which some people consider an organic molecule, but you might remember from Core 1 that to be considered an organic molecule in life, you have to have oxygen combined with that. The second thing that they put in was ammonia. And the third thing was hydrogen gas. All right, so they put all those things in there. They ran an electric current through there. They ran some heat through there. And basically then, and after that, then they condensed the substances out of there using cooler water, and have basically had a tube through here that cool water ran through, and as this stuff dripped through, it would then get cooler, and in those conditions what would happen is you would get a condensation. That is, remember that condensation or dehydration synthesis is how you create polymers. So they actually were able then to get some polymers that resulted from that. the main things that they got were most of the organic molecules and particularly that, and importantly, amino acids and nucleic acids. All right. So those, because you have to have those, remember, amino acids you need to make proteins, that's enzymes that drives a lot of cellular function, and the nucleic acids are part of DNA and RNA. All right, so that was important. The second thing is biosynthesis of monomers into polymers. In other words, they created some monomers, that is the amino acids, the nucleic acids, etc., or nucleotides, I should say, not nucleic acids. They created these monomers. The next thing was to drive or make the polymers. So they theorized that there were different situations maybe that had that involved the evaporation of water. Again, dehydration synthesis that allowed the molecules or the monomers to become polymers or to join them. For example, dripping water on a hot rock might do it. The ocean vents might do it. At this point, I want to mention that you might want to read both the two handouts that I gave you. One looks like this. It's called the Did Life First Appear in the Primitive Pizza? So that's just talking about, you know, some ways. And then the other one was the cartoon history one that I gave you. So just check those out. So once you have some polymers, you have to package that within a membrane. In other words, the membrane, remember, is the biphospholipid layer that's separating one group of water from the inside. And these are called protobionts because at this point they would just be separation of water. But the thing is about that is that if you have any osmotic differences, what's going to happen is that water will move in and out and change the osmoregularity or the osmo solution of that cell, actually, or the protobiont. And then if you had some function of molecules, in other words, if you had things like proteins, you could have the start of chemical reactions that are happening within that cell, okay? And then after that, if you have separate replicating molecules, then you can have the ability of the cell to create another cell and ultimately organisms to create other organisms. All right, so the origin of self-replicating molecules, I think, first started with the RNA because RNA is simpler. It's not a double helix, it's a single helix. And it is a self-replicating, but only really in short enzymes. If you add zinc, it improves the length. So perhaps some sources of zinc or something like that might have been found at the point when the self-replicating molecules started. And you, as long as you had some self-replication happening, then you could have life, basically.